Hello, this is Dr. A again. We're going to cover antimicrobial treatments. All right, so let's talk about some principles of antimicrobial therapy. So, the introduction of modern drugs to control infections was a medical revolution in the 1930s. So, you got to think about this. This is less than 100 years ago. Antimicrobial drugs have reduced the incidence of certain infections, but they have not eradicated infectious diseases and probably never will. Now, when they were first introduced, we thought they were going to eradicate infectious disease, but that just doesn't look like that's going to happen. Today, of course, doctors are worried that we are dangerously close to a post-antibiotic era where the drugs are no longer effective. So, what is the goal of an antimicrobial chemotherapy? So, it's chemotherapy, by the way. I know we have a connotation of thinking of chemo as cancer treatment, but really chemotherapy is taking any kind of chemical as therapy. So, a drug is a chemical. So, therefore, if you're taking an Advil for a headache, it's technically chemotherapy. Okay, so just in this lesson, when we talk about chemotherapy, we're not talking about cancer treatment. We're just talking about taking prescription drugs or over-the-counter drugs to remedy an infection or symptoms and stuff like that. So the goal of an antimicrobial chemotherapy is to administer a drug to an infected person that destroys the infective agent without harming the person or the host cells. So that seems quite logical, but we're going to see as we progress it's not always feasible. So the drug must be able to be easily administered and able to reach the infectious agent anywhere in the body, which is not always possible. For example, it's hard for some drugs to cross the blood-brain barrier, and rightfully so, because it's a way for your body to protect your brain, right, from chemicals and stuff like that. And, they have to be easy to be, taken, whether, of course, the easiest ones are probably the ones that you can take orally, but thinking about something that you have to give IV if you have trouble getting the IV started and that kind of stuff. So, be absolutely toxic to the infectious agent and absolutely non-toxic to the host. That would be ideal. It is not always the case. You can see, just think of even if they're just the approved drugs, the amount of side effects that drugs have, right? And then remain in the body as long as needed, and be safely and easily broken down and excreted. I mean, it does that, which means it doesn't damage your liver, doesn't damage your kidneys, etc. Okay, so let's look at a little bit of terminology here. So a chemotherapeutic drug is simply any chemical used in the treatment, relief, or prophylaxis of a disease. So again, taking some Tylenol or whatever is, a chemotherapeutic drug. All right, what is prophylaxis? If you did not know what prophylaxis is the use of a drug to prevent imminent infection of a person at risk. So if you are given a dose of antibiotic before a surgery or before a dental procedure, that is prophylaxis, especially if you don't have a current infection. Now, I get with a dental procedure, maybe you have an abscess and then go ahead and treat you with an antibiotic or something like that beforehand, and then they're going to go and, fix it. But the idea of prophylaxis is you don't have the infection, but there is a high chance that you're going to have one soon, and because of a procedure usually, so they go ahead and pre-treat you with an antibiotic before the procedure. All right. Antimicrobial chemotherapy is the use of a chemotherapeutic drug to control infection, more specifically. Obviously, there are chemotherapeutic drugs that do not control infections, such as, something you would take for, again, a headache, for congestion, cough, and that kind of stuff. All right. Antimicrobials are an all-inclusive term for any antimicrobial drug, regardless of its origin. So it's a very big umbrella term, whether the drug can be used against bacteria or fungi, whether it is natural or synthetic antimicrobial. If it can work against microbes, it's an antimicrobial. Okay, antibiotics are substances that are produced by the natural metabolic processes of some microorganisms and that can inhibit and destroy other microorganisms. So this is really where we get started with antibiotics. We actually were getting them from other organisms and stuff, and we're going to look at that. And it came from nature, okay? And they were already secreted by some organisms to basically allow them to compete with other organisms and keep these other organisms away from their food. A semi-synthetic drug is a drug that has been chemically modified in the lab after being isolated from natural sources. So we take what we found in nature and we modified it and improved upon it. And a synthetic drug is a drug that is produced entirely by chemical reaction, so it's entirely produced in the lab and it does not have a natural source. And then we have narrow spectrum, also known as limited spectrum, as a definition. So it's an antimicrobial that is effective against a limited array of microbial types. For example, a drug that is effective only against gram-positive bacteria. A broad spectrum or extended spectrum antimicrobial is an antimicrobial that is effective against a wide variety of microbial types. For example, a drug effective against both gram-positive and gram-negative bacteria. So this gives you some of these definitions, and we'll be using these throughout the lesson. So think about it. So when we talked about the ideal antimicrobial, which of the following antibiotic modes of action would have the least toxic effect on a human cell? So the way you want to think about that is the way for it to be the least toxic is it needs to target something that a bacteria has that a human cell does not have. So then look in this list and pick what you think is correct. All right, so let's dig into the origin of these antimicrobial drugs. So antibiotics are common metabolic products of aerobic bacteria and fungi that are produced to inhibit the growth of competing microbes in the same habitat. So it's to compete for food and resources, okay? So generally speaking, we have been getting these from bacteria in the genera Streptomyces and Bacillus, and molds in the genera Penicillium and Cephalosporium. So this plate will give you an example. You have the mold Penicillium here that is growing, and then you have a culture of staph that had been inoculated, you see, all the way across, right? And then they drop the Penicillium mold right there. And where the Penicillium mold is growing, it's secreting some antimicrobial, some chemicals that are killing the staph bacteria, and so it's inhibiting the staph from growing, and therefore the penicillium has access to the food in this clear area here, and the staph cannot get to it. And so it actually even killed and inhibited all the bacteria that would have been in this zone right here, right there. Okay, so identify the agent and starting treatment. This is going to be very important for you guys to really latch on to. If most of you guys, I imagine, is going into nursing or some kind of medical practice. And so it's really important to understand that before the actual antimicrobial therapy can begin, there's 3 factors that have to be in place. First, you have to know the nature of the microorganism causing the infection. So ideally, you would have some preliminary results, some culture results, or something. Obviously, a lot of times, just in order to not delay treatments, physicians will basically do a best guess and go ahead and start some treatments. And so one way you could know would be if a patient came in the ER and they did a rapid strep test and it was positive, then you know for sure this patient has strep and you can start them on a specific treatment for that. And if you did a UA and you see urinalysis and you see a bunch of bacteria, and there are some likely culprits like E. coli that the doctor could assume that is going to be this type of bacteria, and so they'll prescribe an antibiotic for that sometimes without even specifically identifying it. And if they order a culture that's going to take a few days, so let's say a sputum culture or blood culture, It has to be plated and grown out and identified. What they will do is they will collect the specimen before you want to collect the specimen before you start the treatment, and go ahead and start the treatment on the presumption of a certain organism that is likely to be causing the symptoms, but awaiting the culture results and potentially changing the treatment when the culture results come in. Okay. Another factor is the degree of the organism's susceptibility or sensitivity to various drugs. So that would be the difference, for example, of having a regular staph infection in a wound and having an MRSA staph infection. So one would be more, this just regular staph infection would be more easily treated with just some run-of-the-mill antibiotics, whereas the MRSA, you would need some more, just some stronger, probably IV antibiotics to knock it out. And so when we get a patient that has a wound or something that could potentially have a microorganism that is resistant, it is important to get not only the identification of the microorganisms, but how sensitive it is to various antimicrobials. So we're gonna talk about that here pretty soon. And then we also need to consider the overall medical condition of the patient. Is this patient young and otherwise healthy, with good liver function, good kidney function, and all of that, so they have a good ability to metabolize the drug and clear it? Or is this patient that is on multiple drugs with existing conditions that could make the infections worse, with compromised renal function? Just think of a type 2 diabetic patient that's already, maybe in the advanced stages of type 2 diabetes, which diabetes makes any kind of infection worse. Their circulation is not that great. Their kidney function's usually not that great and stuff. And so they could be a little bit more challenging there in their treatment. So identification of the infectious agent should begin as soon as possible. And I cannot overstress this. And ideally, you should get your cultures before the antimicrobial drugs are given, therefore before their numbers are reduced, because as soon as you start treatment, hopefully treatment is going to work and start knocking down the bacteria or the microbes that are growing. And if it is really effective, then you might miss your opportunity to correctly identify what was infecting the patient. Now, is that always relevant to clinical practice? No. There's sometimes, again, the doctors will just make an assumption, for example, again, on a young, healthy, I'll say female with a urinary tract infection. They may not bother to actually get an identification unless the treatment is not working. All right. But ideally, you want to get your samples, you want to get your cultures at least collected before you hang those antibiotics, before you start dosing them with antibiotics. The direct examination of sample is the rapid method for detection. So that would be, for example, your rapid streps, and there are other tests that can be done that are very similar. So you test for flu, you test for RSV, which those are viruses, and they do have some antimicrobials that are used for them, but not all of them do. And then you test for C. diff and other things like that. We can actually use a test that you use the sample, the swab itself, or the poop sample, or whatever, and you can get a positive, negative. You can know really quickly. Doctors often begin therapy on the basis of immediate findings and informed guesses. So again, like I stated, they, whatever test they can do. One of the big ones in the lab is to do a CBC, a complete blood count. And from the results of that, you can see indications if the patient has a bacterial infection versus potentially a viral infection. And so that can give you some clues, give the physician clues as to what's going on with your patient, in addition to the signs and symptoms, and then, the quick tests. And sometimes epidemiological statistics may be required. So what do we mean by that? Well, for example, currently with the pandemic, where the coronavirus is currently, like what's the average number of cases in the area, is this something that is occurring in this area or not, et cetera, et cetera. And so those can be relevant there too. So like now, because we're in the middle of the pandemic, if a patient comes with symptoms XYZ, then they would run a coronavirus test, whereas a year ago that would not have been even thought of, right? So let's talk about testing for drug susceptibility. So there are certain organisms where it is necessary to test to see what antibiotic they are susceptible to, because there are different strains that are resistant, and they can be a challenge sometimes to treat. So any Staphylococcus species, Neisseria gonorrhea, any Streptococcus species, and or genera. So Staphylococcus species, Streptococcus species, Neisseria gonorrhea, and then all your aerobic gram-negative intestinal bacilli. All of those you have to get drug susceptibility testing. And testing for fungal or protozoal infections is difficult and unnecessary. We just can't. One is just it's not possible to do. Um, there are some advances we've done in the genetic testing and genetic profiling of these microorganisms, where it could be possible to get the genetic profile of a resistant fungi or protozoan and ID it that way. But this is some of the pretty advanced testing. And the traditional microbiology testing for drug susceptibility, we just don't do it on fungi and protozoans. And part of that, as you're going to see, is we have a limited arsenal against these guys anyway. So I mean, your choices of drugs are limited. So you're just going to start with what you have anyway. Okay, so we can all go back over several cases that we've already looked at. So this one was a 24-year-old female automotive technician that had fever and pain in her left hand. She had that wound in her left hand with the purplish red streaks running up her left arm. She had enlarged lymph nodes at the elbow and her arm, and her skin was warm and dry. And so she, if you remember, had an infection. So this is what grew from hers. Again, we covered this a few lessons ago, right? So it was some gram-positive cocci in clusters that grew in that wound. And do you remember what that would be, gram-positive cocci in clusters? Which of these organisms might it be? Now, that's bacteria. These are all bacteria. So just, again, see if you remember what it was, right? And then we're going to talk a little bit about testing for drug susceptibility. So there are a couple of ways to do that. The first technique that was used, and it's still in existence today, we can still use this technique for certain organisms, is the Kirby-Bauer technique. And so the way we do this one is we take an agar plate that has plenty of the food for that specific bacteria, so something that bacteria will love to eat and grow on. And we actually, instead of doing the streaking for isolation, we actually just cover the whole surface nice and evenly with bacteria. So the entire surface of the plate is covered with bacteria. So they'll grow kind of diffusely across the whole plate. And then what we do is, right after we spread all the bacteria all over, so you don't see any growth yet, is you drop discs of antibiotic all over the plate. And often these plates are done, they're actually quite big. Okay, so we can do quite a few antibiotics, and we have to spread out these discs around. And then around each of these antibiotic discs, if the antibiotic is effective, there will be a zone of inhibition, a zone of no growth. So the bacteria that has been put on the plate will be able to grow up to a certain area close to that antibiotic. And then you can measure that zone of inhibition, so that zone of no growth around that antibiotic disc, and compare it to a standard for each drug. And then from that, you get an antibiogram, and that can give you information for proper drug selection. This method is less effective for anaerobic bacteria, for fastidious or hard to grow bacteria, or for slow growing bacteria, because it would just take you forever to get some results. So this is what it would look like. So again, you have the plate. You've inoculated all the way across with bacteria. So bacteria grow all over. So this, all the light yellow color is bacterial colonies growing. And then you have each disc has been dropped here. They have an abbreviation on there denoting what it is. So, for example, OT is oxytetracycline, and it's 30 because it's a 30-gram dose. And then we have GN is gentamicin, etc. And so you have each one of these, and you can measure across the diameter. So you go from one side across the tablet to the other side and measure it in millimeters. Okay, so you measure that zone. It should be equal all the way around, and you do it for all of these, right? So this is a photo of what it would actually look like. And sometimes those zones do run together. So if you need to measure it, then you're going to measure it kind of across this way or maybe across that way, right? So you can get from one side to the next, from one side to the next right there. And you record that. And then there is a table, database and stuff of what would be considered resistant, sensitive, or intermediate. And so it would be something like this. So, for example, this is an example for Staph aureus. So again, that is what that patient had with her in her wound, in her hand wound. She had Staph aureus growing. So if this was her Kirby-Bauer plates and her antibiogram results, then, for example, for bacitracin, in order for it to be this bacteria to be susceptible to bacitracin, the diameter must be greater than 13 millimeters, and if it is less than 8, it is considered resistant. The diameter was 15, therefore it is sensitive. For erythromycin, for it to be susceptible, it has to be greater than 18. If it's resistant, it has to be less than 13 millimeters. We got a 15, so it's kind of an in-between, so we're going to put intermediate. So probably not going to be your best choice. You'd probably want to go with one where it says sensitive, right? You know, because there's a chance it may not work properly there. And then, for example, you would not want to pick neomycin, which is, by the way, what's in triple antibiotic, or polymyxin B, or penicillin G, or anything like that. So, because you need, for example, for neomycin, you would need to have a zone greater than 17, and if it's less than 12, is resistant. So since it is 12, we're going to consider it resistant. Penicillin needs to be greater than 29. It cannot be, if it's less than 20, it's resistant, and this portal thing was only 10, so it's definitely resistant. And so that might be why the antibiotic cream or whatever she had put in it did not work, right? So all of these are resistant, so you would not select any of these as potential therapy, but you could select vancomycin or tetracycline because these are sensitive, or you could select kanamycin, gentamicin, chloramphenicol, or bacitracin to treat this patient. So normally in the lab, we would do our antibiotic sensitivity lab. So you have that as an assignment in your labs. And so if hopefully you view this, and then after that you do your lab, and then that will make sense and kind of explain what's going on with the Kirby-Bauer. So you have a Kirby-Bauer plate that you picture of that you're going to use to estimate the sizes of the zones of inhibition and pick the best antibiotic. So the goal of an antimicrobial drug, of course, is to disrupt cell processes or structures of bacteria, fungi, or protozoans, or to inhibit virus replications, right, because they're not a cell, to interfere with the function of enzymes that are required to synthesize or assemble various macromolecules that are either needed for the viruses or the bacteria, etc., or destroy structures already formed in the cell. And so these are all different ways that these guys can work, and they need to be, again, selectively toxic, meaning killing or inhibiting the microorganisms, but not killing our cells, our vertebrate cells. So we want it to destroy the bacteria, but not destroy our cells. So drugs that have excellent selective toxicity block the synthesis of bacterial cell walls. The original ones were the penicillins for that. Why does that have excellent selective toxicity? So selective toxicity, again, is being toxic to the bacteria or the microbes and not toxic to us. is because none of our cells have cell walls. We have cell membranes, but we do not have cell walls. So that's something that's very typical of bacteria. And so it's good to use those because they'll work on bacteria, but they don't cause harm to our cells. So human cells lack the chemical peptidoglycan. We just don't use it. That is peptidoglycan, if you remember, is part of what cell wall, especially gram-positive cell walls, are made of. And therefore, we are unaffected by the drug because we don't use peptidoglycan. The drugs that are most toxic to humans are drugs that act upon a structure that is common to both the infective agent and the host cell. For example, if it acts on the cell membrane, it could also disrupt the cell membranes of your cells. And as characteristics of the infectious agent are more and more similar to the host cell, selective toxicity becomes more difficult to achieve. So if you think about that, we've looked at bacteria, which are prokaryotes. We've looked at fungi and protozoans and all that, which are eukaryotes, and we've looked at viruses and stuff like that. Of those three, the ones that the prokaryotes, I'm sorry, you have the eukaryotes ones, so the fungi, protozoans, and helminths, those the eukaryotic cells are very, very similar to our cells because our cells are eukaryotic cells, right? So the structure of a fungal cell is very similar to the structure of a human cell. And so it is really hard with antifungal agents to achieve selective toxicity. Okay, so again, the goal of chemotherapy is identifying structural and metabolic needs of a living cell and then removing, disrupting, or interfering with these requirements. And so when we're talking about the living cell, then we're talking about those bacteria, fungi, protozoans, and stuff like that. So we want to target something of that cell that we're trying to get rid of, that bacteria or fungi that we're trying to get rid of, and just basically take all its food away and starve it, or break down its cell walls, or poke holes in its cell membranes, or something like that. So your antimicrobials have several big drug categories, okay? There are a bunch that inhibit cell wall synthesis. Again, great target because we don't have cell wall. There are some that inhibit the function of, the structure and function of nucleic acids, so they target DNA and RNA, okay? So there's less in those, of course, because it's harder to achieve selective toxicity. And there are some that inhibit protein synthesis. Those, again, there's a few of them, and they're especially helpful because the ribosomes of prokaryotes are different than the ribosomes of eukaryotes, so it's possible to target some of those differences. And then there are some that interfere with the cell membrane structure and function. Again, harder to achieve selective toxicity. And then there's some that inhibit folic acid synthesis, which could also mess with your folic acid synthesis. It's totally possible. So this diagram has kind of the main ones, and so we'll start with your cell wall inhibitors, right? So those the antimicrobials that target the cell wall. So they block synthesis and repair. So those are your penicillins, cephalosporins, carbapenems, vancomycin, bacitracin, fosfomycin, and isoniazid. The ones that target the cytoplasmic membrane and they cause loss of selective permeability are your polymyxins and daptomycin. The ones that mess with the DNA, RNA synthesis or function, so some inhibit the replication and transcription. They inhibit gyrase or your quinolones. And the ones that inhibit RNA polymerase are rifampin. The ones that inhibit folic acid synthesis in the cytoplasm are your sulfa drugs and trimethoprim. So, sulfonamides and trimethoprim here. And then the ones that act on protein synthesis, again, they act on the ribosomes, which prokaryotic ribosomes are different than eukaryotic ribosomes. So the ones that work on the 50S subunit are erythromycin, clindamycin, synarcid, and pluromulins. Those mutulins. Anyway, I can hardly read that one. The 30S subunit are your aminoglycosides like gentamicin and streptomycin, your tetracyclines and your glycocyclines. and those that target both 30 and 50S is linezolid. There you go. So you kind of have how these guys are divided up. Okay, so what class of antibiotics would work best on gram-positive cell walls? So remember, gram-positive cell walls, what are they made of? They have a really thick layer, right? And so thinking about that, which of these would work on the best on gram-positive cell walls? Okay, so again, our patient in our case that we brought back up had staph. Staph is a gram-positive bacteria. And so we would use drugs that target cell walls. So some of the ones that are starred are the ones that would work. So you could select a penicillin, or you could select a cephalosporin. Now, we saw on the antibiogram that it was resistant to penicillin, so you probably wouldn't. But if it was sensitive, you could select penicillins, technically work against those. It's just that bacterial species do adapt. Penicillins are some of the oldest drugs, so they've had the most time to adapt to those. And so we have quite a few that have developed roundabout mechanisms to inactivate the penicillins and work around them. And so that's why we've had to develop new drugs and stuff. So, and if you look here, you have the natural ones, and then the ones listed after that are the ones they started modifying in the lab. So your penicillin G and penicillin V are your most natural forms, and they're natural, and they treat gram-positive cocci and a few gram-negative bacteria. Then you've got ampicillin, carbicillin, and amoxicillin. They have a broader spectrum of action and are semi-synthetic. Again, so we started messing with them in the lab. And then the next generation of those is methicillin, niacillin, and fluoxacillin. And again, they are semi-synthetic, and they are some of the first ones that were trying to bypass the penicillinase producing bacteria. So penicillinase is an enzyme that can inactivate the beta-lactam ring in penicillins and cause them to be inactive. And then from there we got a few others, and that were, again, some of them are combinations, some of them are another generation trying to inhibit the beta-lactamase enzymes. Then you have your cephalosporins, and they're listed again, first generation, second generation, third generation. So first generation is the first ones used, probably the most natural ones, cephalothin and cefazolin. But then they started modifying them in the lab, so some of these are going to be semi-synthetic and stuff. And so, each improvement makes it a different generation of drugs. So we have cefaclor is your second generation, cephalexin is your third generation, ceftriaxone is another third generation semi-synthetic, broad spectrum. And then we got cefpimine, and can hardly read it. They're fourth generation and fifth generation ones there. And these were still targeting cell walls, and you have your carbapenems, which you could use also. And you have imipenem and aztreonam. So these guys are powerful but potentially toxic, and usually reserved when other drugs are not effective. And aztreonam is narrow spectrum and is used to treat gram-negative aerobic bacilli causing pneumonia, septicemia, and urinary tract infections. Effective, but again, for those who are allergic to penicillins. And then the miscellaneous drugs that target cell walls, we have bacitracin. Again, this one's narrow spectrum, mostly used for skin infections, for strep and staph. It's the main ingredient in Neosporin, triple antibiotic ointment. Isoniazid is used to treat Mycobacterium tuberculosis, but is only against growing cells. Vancomycin, so this one was a big gun, is narrow spectrum, but it's really good for staph infections, especially MRSA, and or patients are allergic to penicillins. And fosfomycin, and it's a phosphoric acid agent, effective as an alternative treatment for urinary tract infections caused by enteric bacteria, in case they're allergic to like the sulfa drugs or something. Okay, so let's go to an open-ended question. Our patient's infection got worse and not better. You suspect that she has MRSA. How does MRSA happen? So how do you get antibiotic resistance? So put any of the mechanisms that could cause MRSA or just that can cause antibiotic resistance there. You're going to have some assignments on antibiotic resistance in this unit anyway. So drug resistance is an adaptive response in which microorganisms begin to tolerate an amount of the drug that would normally be inhibitory or be killing them, right? So they want to survive, and so they have ways to mount defenses and adapt to environment where that specific drug is present so that they can survive. So drug resistance is due to the genetic versatility and the adaptability of microbial populations. Of course, if you look at, the way they grow, I mean, they get a new generation, some of them every 30 minutes. And so it is quick to adapt and turn certain genes on and off and then pass those down. So anyway, because they have this short generation time. And some of the drug resistance mechanism can be intrinsic, so it's built in. So it's a gene that they already have, and it just won't turn on if it's not needed. But as soon as you expose them to the drug and they need to survive, they'll turn that gene on, which will allow them to survive. And then it can be acquired. We're going to cover how those can be acquired. So chromosomal drug resistance. So it's usually from spontaneous random mutations that allow them to have a gene now that works as a resistant gene, a resistance mechanism against a specific drug. Slight changes in drug sensitivity can be overcome with larger doses of the drug if you're, treating a drug-resistant organism. Persisters are organisms that slow or stop the metabolism so that the microbe can't be harmed by the antibiotic. So basically, they quit taking in anything into the cell or whatever. So it's almost like, if you will, they quit eating so they are not going to be poisoned. And that allows them to persist and stay alive. So let's talk about horizontal transfer. So first of all, let me talk vertical transfer. It would be transfer of genetic material from parent to offspring. So in the case of a bacteria, one parent cell will divide up and form two daughter cells, and they will have identical genetic, profile. And so the two daughter cells would have the same genes. And if the parent cell was resistant, then the two daughter cells would be resistant, and then they will have, split in two, and then so the gene keeps moving. So that's vertical transfer. Horizontal transfer is when one bacteria shares a resistant gene, a resistant mechanism with its buddy, not through, the replication or anything like that, not through division, binary fission, or making offsprings. So a resistance factor is a plasmid that contains an antibiotic resistant gene. So plasmids are bits of DNA code that exist separate from their genome, from their traditional genome. So you have all the chromosome material here, and then you have the plasmid over here, and it's just one single little gene all by itself. Think about it this way: if the genetic material was a cookbook, and if you had the cookbook here, and it had all the genetic code, it had all the recipes, right? And then you had a little recipe on a note card over there. The plasmid is the recipe on the note card. Okay. So how then do they get this recipe from one cell to another cell? So there are three mechanisms. There's conjugation, transformation, and transduction. Okay. So and we're gonna cover all three. Plasmids encoded with drug resistance are naturally present in microbes before they have been exposed to an antibiotic. Again, so that gene could just be there, and the fact that they are exposed to the antibiotic activates that genes. Transposons also can duplicate and insert genes for drug resistance into plasmids. So that's the molecular machinery that can put those in there. They're called transposons. And sharing of resistant genes accounts for the proliferation of these drug-resistant species. So the deal with this is not only could, like, for example, one staph bacteria share the code with another staph bacteria that's not its offspring, right? It can also share that code with, let's say, a strep bacteria or an E. coli or something like that. So it can share across species. So, development of resistance and its long-term therapeutic consequences. So, any large population of microbes will always contain a few cells that have drug resistance, but you won't know it unless you give antibiotics. So, as long as the drug is not present in the habitat, the genes are not expressed. You just don't know it. If the population is exposed to drugs, so therefore patients taking a drug, the drug-resistant population then has a selective advantage, meaning that all the microorganisms that don't have the drug resistance genes are going to be eliminated, and therefore the ones that have the drug resistance genes have free reign to all the resources and can multiply quickly without any competition. And of course, then they can multiply and make more offsprings, and all their offsprings are going to have the drug resistance gene in them. And the replacement population, which is now the resistant one, will have the drug-resistant form as the dominant species because all the ones that were non-resistant got eliminated by the drug. Okay, a few new approaches to antimicrobial therapy. You can target iron scavenging capabilities of bacteria. So bacteria need iron to duplicate. It's like a food for them. And so, which is an interesting thing, if you ever check iron levels on somebody that has a bacterial infection that's raging, that you'll see the iron levels are going to be really, really low because your body will hide that iron from the bacteria to try to attempt to prevent them from growing, right? There's RNA interference strategies, or RNAi or RNA molecules that are designed to cut up and chop up RNA and DNA sequences that are found in a cell or in the body that don't belong. Okay, and so that's one way you can mess around with it. You can mimic defense peptide molecules. So our body, part of our immune system, does produce certain defense molecules, and so we can replicate those and use those. Are kind of expensive. And using bacteriophages. So this little fancy guy right here is a bacteriophage. It looks like a little spacecraft, right? So what is a bacteriophage? Well, as you recognize here, this is a icosahedron. So guess what? That is a virus. It has DNA or RNA material in here, never both, right? Because it's a virus. And it can actually dock its little landing things here on the cell surface of a bacteria, and it can insert its genetic material. It just pokes it in there and can insert its genetic material inside the bacteria. And so bacteriophages are viruses that only infect bacteria, and they're very specific. So for example, a bacteriophage that would infect staph would not infect E. coli or would not infect strep. So, and I would say even one that affects Staph aureus may not affect, for example, Staph epidermidis. And they will, what they do is they infect the bacteria, and they do the same thing that viruses do to our cells. If they can enter into that cell and put their genetic material, it's just going to take over that bacteria, make a bunch more bacteriophages, which will cause the cell to rupture, release all the bacteriophages, all the viruses, and then the viruses are going to go and find more bacteria to infect and keep repeating the process. Okay, so we're like, ooh, okay, well, we could use this process to help fight infections. And so this has been used in Europe some, and it's been used also in some of the food production and stuff to limit spoilage. Is basically you purposely infect a person with bacteriophages that are targeted to the organism that is causing the bacterial infection. So you basically you inoculate him with a bacteriophage virus to treat a bacterial infection, knowing that the bacteriophage viruses are only going to target that specific type of bacteria and then will not do any harm to any of the human cells. So that's really an interesting thing. So this other case that we saw, so remember, it was a 5-year-old in general health and condition, but he was presented because he had a fever. He was cranky, had a sore throat for about 24 hours. The patient had a fever of 39 Celsius and had considerable swelling and drainage of the pharynx and conjunctivae, as tonsils were large and coated with white patchy exudate right here. Had a red throat and swollen anterior cervical lymph nodes. His ears were clear. His chest sounded clear. He had no additional remarkable findings. So if you remember that case, this little guy had gram-positive cocci in chains, also known as streptococci. So, of course, you should be able to pick out what was wrong with him here. Again, all of these could cause problems in the throat, right? So we have diphtheria, influenza, RSV, and strep. And so hopefully you guessed correctly, right? And so with this, we're going to talk about a second method to look at antibiotic sensitivity. And this is called a tube dilution test. So this is, again, even though we still can use Kirby-Bauer on some microorganisms, this is more the standard of how we test for drug susceptibility in a lab now. Other than now, it also has, there's the genetic profiles. And so we're using genetics now also to test for drug susceptibility. So we're looking at in their genetic profile of microorganisms to see if they have certain drug resistance genes and stuff. But so let's get back to the tube dilution test. So it is more sensitive and quantitative than the Kirby-Bauer test. So we always like that better. More sensitive, so it can pick up more minute differences and stuff, and can, you know, it's easier to pinpoint, right? And quantitative, you can get actual measurements there. And antimicrobial is diluted serially in tubes of broth. We have miniaturized it into microwell tubes, right? And then each tube is inoculated with a small uniform sample of the pure culture of the bacteria that you're trying to test. So in this case, with a little boy with swollen throat who has strep throat, we would take some of that strep bacteria, make a suspension, and put an equal amount of that suspension all the way across all the tubes, okay? And then you put the antibiotic. The antibiotic is diluted serially from I believe most concentrated to least concentrated. I'll show you in the so it goes one way, and we put the same amount of bacteria all the way across. And then we look for the minimum inhibitory concentration. So it's the smallest concentration or the highest dilution of the drug that will inhibit the growth of that microorganism. And it's very useful in determining an effective dosage and also providing a comparative index against other microbials. So this is what it would look like. Okay, so again, you have the same inoculum size of bacteria added to all the tubes, so all the way across here, and these are mini tubes and mini well plate, right? So the negative control, and here's a negative control, there's a positive growth control here. This is just a control to make sure that the bacteria actually did grow. And then from here on out, you have different concentrations, right? And where it's increasingly concentrated. So the most diluted is here, the most concentrated is here, okay? And so you can see here, if it is pink, it's the bacterial colony, it's the bacteria growing really well. And if it is purple, there's no bacteria growing in here, okay? And so for this, I think it's probably amoxicillin and fluconazole and everything like that. We have, so this abbreviation for this antibiotic, we have these dilutions, so more diluted, less diluted, less diluted, less diluted. All of these bacteria grow, and so this would be the MIC at this dilution right here, and then all of these are a little bit more concentrated, a little bit more concentrated, a little bit more concentrated, more concentrated. This is the lowest dilution, right, that inhibits the bacterial growth. So this would be the ideal one, because you don't want to give too much of a drug. So if giving this amount would work. This one's the MIC for that one, for this one, and then you can go and see. And then so you can generate a report and you can compare them. And so a little bit about MIC and the therapeutic index. So there's one thing that we have to recognize is in vitro activity of a drug is not always correlated with the in vivo effect. So what the heck does that mean? In vitro means in glass, which means in the test tube. So the what you do in the lab doesn't always correlate to what happens in the body. And in the body is in vivo, vivo for alive. So in vitro in the test tube, in vivo in the patient's body. Okay. Why is that? So it could be that the drug cannot diffuse into that body compartment. So yes, technically it would kill it, but it cannot get to where it's at, to the brain, joints, skin, or whatever. Or there were resistant microbes in the infection that didn't make it into the sample that was collected for testing. So it's a factor that's not accounted for. Or the infection is caused by more than one pathogen, which is so mixed pathogens, some of which are resistant to the treatment, right? So those are all possibilities. And so we've also developed what we call the therapeutic index. So the therapeutic index is the ratio of the dose of the drug that is toxic to humans as compared to its minimum effective dose. So we're looking at the dilution, the MIC that is effective, right, just for the bacteria, comparing to how harmful that dosage is to human tissue, right? So the smaller the ratio, the greater potential for a drug reaction or for harm. Okay. So a therapeutic index of 1 or 1.1 is really risky, is really dangerous, high potential for harm to the patient. But a therapeutic index of 10, which is 10 times more, is a much safer choice. So the higher the number, the safer the choice is. And so with your antibiogram there, with your MIC, you get your MIC for all the drugs, but then you also get a therapeutic index for all the drugs. So you can, with that, using that information, you can find one that is both safe and effective. And so that allows you to select the most safe and effective treatment for your patients. So again, highest therapeutic index has the widest margin of safety. And so, again, the physician has to take careful history before prescribing an antibiotic. So are there any pre-existing conditions that might influence the activity of the drug? For example, well, even like, are they on another drug that could interfere with that drug? Do they have an allergy for that drug or something like that? Which that was the second one, history of allergy to certain classes of drugs, right? Do they have a, any condition that would interfere with the metabolism of that drug, the distribution of that drug, or anything like that? Are there any kind of underlying liver or kidney disease? Why are liver and kidney disease important? The liver detoxifies the drug and clears it from the body, and so does the kidney. So proper liver and kidney function are essential for the drug to clear the body. Infants, elderly, and pregnant women also require special precautions. So infant, because they're so small, it's easy to overdose them. The elderly, because they're more frail and their kidney function has declined in most elderly people already. And so usually they're already often on other drugs, so they just take a little more careful consideration. And then pregnant women, of course, because some of these drugs could be harmful to the baby, to fetus. So again, intake of other drugs can result in increased toxicity or failure of one or more of the drugs. So it could be, for example, did you know like birth control pills would be canceled out by an antibiotic? So if you're on birth control pills and you take an antibiotic, the antibiotic can cause the birth control pills to not work, and then you become pregnant. So that's a drug interference. And the certain drugs are metabolized by a specific liver enzyme, and if you're taking more than one drug that's metabolized by the same enzyme, it could slow the metabolism and clearance of that drug. And of course, any genetic or metabolic abnormalities. So, maybe you don't have enough of those enzymes that clear that drug or something like that. And of course, the site of infection, route of administration, costs, etc. So, are you trying to reach an area that's kind of harder to reach? The route of administration, can you give this drug orally, or do you have to give it IV? So think about if your patient was in ICU and on the ventilator and was NPO. So automatically, like, they're not taking anything by mouth, even if they have, let's say, a feeding tube and all of that. You can't, maybe you just can't give them anything by mouth. They have a tube in their throat. They can't swallow anything, obviously. Therefore, you would automatically have to select something that's either injectable IM or something that's IV, right? And then the cost. There are some antibiotics where you go fill a prescription and it'll cost you, like, less than $10, sometimes even just a few dollars. And there's some antibiotics that cost like $20 a pill, and one prescription might cost you $200. So that is also something that physicians should take in consideration. Okay, which of the following antibiotics would be the safest choice for a patient with no exceptional medical history? So just, average patient, good health. So drug A has a zone of inhibition of 30 millimeters, a therapeutic index of 1.2. Drug B has a zone of inhibition of 12 millimeters and therapeutic index of 12. So assume that both of those zones of inhibition are effective and are, the drug, the bacteria is sensitive to that specific drug. Because I know you don't have the tables. Okay, so our case, that was strep throat. They are the types of antibiotics, again, because it's a gram-positive, would be some that target the cell wall. So for strep, penicillin, G and V are good. Your ampicillin, amoxicillin, and all of that could be effective against strep. And then the other ones are some of these that we haven't looked at are the drugs that target protein synthesis. So in those, we have tetracyclines. They block the attachment of transfer RNA to the receptor, and so it stops protein synthesis. So we have your tetracycline and terramycin are here, and they're effective against gram-positive and gram-negative rods and cocci, and aerobic and anaerobic bacteria. So they're pretty good there, and also Mycoplasma rickettsia and spirochetes. And then you have your glycocyclines like tigercycline, and it's a newer derivative of tetracycline. And again, they're effective against bacteria that are resistant to tetracycline. And my husband had to have a treatment. He was hospitalized. He had to have some tigercycline. It is not fun, and it was expensive. Macrolides are your next going to be. This is going to be another treatment of choice for streptomycin. So this is your erythromycin, clarithromycin, and azithromycin. So I believe it would be your Z-Paks there. So they're relatively broad spectrum, semi-synthetic, and used in treating ear, respiratory, and skin infections, as well as Mycobacterium infections in AIDS patients. And then a few miscellaneous drugs that target protein synthesis. We have clindamycin, quinaprestin, and lisinozid here. So let's look at two hypothetical cases, and I want you to think of what could be a problem in these cases. So if you have an elderly alcoholic patient with pneumonia that's caused by Klebsiella and is complicated by diminished liver and kidney function that requires parenteral or IV drug administration, and she has a history of allergy to penicillins. Okay, so which factors in all the description here would cause you concern, or would be things that you need to take in consideration? The next one is a cancer patient with severe systemic candida infection. So what are the considerations in this one? So again, even when all the information is in, the final choice of a drug is not always easy or straightforward, and that's why physicians are paid the big bucks and carry malpractice insurance, honestly. So in here, I want you to put which are the factors that you need to consider in this scenario with the elderly alcoholic patient with pneumonia. And then in this one, I want you to put what factors need to be considered in your cancer patient. Okay, so again, a little bit on spectrum of activity. Broad spectrum, or effective against more than one group of bacteria, like tetracyclines. Narrow spectrum, target a specific group, like your polymyxins. And your penicillins, for example, the original one was narrow spectrum, and susceptible to microbial counterattacks with the beta lactamases and stuff. But the molecule was altered and improved upon over the years with each successive generation with laboratory modification. And so the later penicillins overcome the limitations of the original molecule. And so here you have a representation of, what really a visual of broad spectrum versus narrow spectrum. So you see polymyxin, only effective on some gram-negative bacteria. So is tobramycin on TB and a few gram-negative bacteria. But then you look at tetracyclines, and bam, there are like 1, 2, 3, 4 things that can go. Some gram-negative bacteria, gram-positive bacteria, the chlamydias and the rickettsias, which cause Rocky Mountain spotted fever. And so it's really interesting to see. And so if you have gram-positives, you'd be selecting from these. Gram-negative, you can select from these. TB, you can select from these. But it gives you kind of a good visual. And again, this is more of a breakdown of the different generations of penicillin and what the advantages and disadvantages were. So if you looked at the original penicillin G, here's the beta-lactam ring in penicillin G. It was the best drug of choice when bacteria are sensitive because it's low cost and low toxicity. So excellent choice. But because that beta-lactam ring can be hydrolyzed by penicillinase, then it can be ineffective. You can have allergies, and it does require injection. So there's advantages and disadvantages. But then if you go down here, for example, and it was narrow spectrum, now ampicillin, for example, or let's just do amoxicillin is broad spectrum. The advantage is you can use it now against gram-negative infections. It has good absorption, but the disadvantage is some people are allergic to it, and it can be hydrolyzed by penicillinase and stuff. So you can explore that in your book. So let's look at this 25-year-old woman that goes to her family physician complaining of itching and irritation down there with burning sensation while urinating and a thick, white, odor-free vaginal discharge. She recently had a strep throat infection and was on a course of Cephalexin antibiotics, which she had just completed. So, again, this is a case we've looked at in our eukaryotic chapter. She has budding yeast, and it is candida, which basically means she has a yeast infection, right? So as a quick reminder, do you remember which one she has? You should be able to select that since I just told you. And so let's talk about agents to treat fungal infections. So eukaryotic cells that present special problems in chemotherapy, the problem is they're eukaryotic cells, and our cells are eukaryotic cells. Therefore, they're just more challenging to treat, period. Okay. So all the drugs that are designed to act on bacteria, which are prokaryotic cells, are ineffective against fungi because they're eukaryotic cells. They have different ribosomes. They have just different everything, right? And the similarities, obviously, between the fungal and the human cells means that the drugs that's toxic to fungi will harm human tissues. So that's a problem. And there are only a few agents with special antifungal properties have been developed. Okay, so here they are. Okay, that is it. This is all we have to treat fungal infections. So we have macrolide polyenes. So these are your amphotericin B. And they bind to fungal membranes, and it can be used to treat skin mucous membrane lesions caused by Candida albicans. And there is an injectable form of the drug that can be used to treat histoplasma and cryptococcal meningitis. But it is rather toxic to the person taking it. Then you have all the azoles. Anything that in an azole is an antifungal. So you have ketoconazole, fluconazole, miconazole, and clotrimazole, and they all interfere with sterol synthesis in the fungi, which that sterol is specific to fungi, and you use them for cutaneous mycosis, vaginal and oral candidiasis, and systemic mycoses. And then you can use them for AIDS-related mycoses, and you can also use them to treat infections in the skin, mouth, and vagina. So again, if you go to Walmart and pick up some, vaginal yeast infection, it's going to be one of those azoles. And if you go to Walmart or CVS or Walgreens or somewhere, some pharmacy, and pick up treatment for athlete's foot, it's going to be, again, a variation, one of these azoles there. Then we have your echinocandins. So you have mycofungin and capsofungin. They inhibit fungal cell wall synthesis. So fungi do have cell walls and human cells don't, so that's a good target there. And they're used against candida strands in aspergillosis. And then you have nucleotide cytosine analogs. So they mess with your nucleotides, your DNA and RNA, and that's flucytosine, and it's rapidly absorbed. It dissolves in the blood and spinal fluid, so it does reach the blood, the brain and through the blood-brain barrier. It can be used to treat cutaneous mycosis, but usually it's combined with amphotericin B to treat systemic mycoses because many fungi are resistant to this drug. So you need a combo with amphotericin B in order to be effective. And so part of, why would you treat it systemically? Because most of our fungal infections we think about thrush, vaginal yeast infections, athlete's foot, ringworm, and things that are just kind of minor. But if you have a chemo patient or patient with cancer and they've had chemotherapy and their immune system has been wiped out, they are very susceptible to these random fungal infections, and those things can go systemic, so they can actually spread throughout their whole body system. They can get in their blood and cause all kinds of problems. A little bit about helping nature along. I really like this. Probiotics and prebiotics. So in this case, with the woman with the vaginal yeast infection, as you remember, she had been on a course of antibiotic for a strep throat infection, and the antibiotic for the strep throat had caused her to lose enough of her normal flora to cause the yeast to grow out of control and give her a vaginal yeast infection. This is something that's really, really common. Well, one of the things you can do to kind of help prevent that is to take probiotics. So probiotics are preparations of live microorganisms that are fed to animals and humans to improve your intestinal biota. So the idea is, and you shouldn't take it at the same time as the antibiotic. You should kind of space them out. So take your antibiotic dose and then later on take the probiotic. Is the idea as the antibiotic destroys some of the good guys, you're replenishing more behind. And so it can either replace the microbes that were lost during antimicrobial therapy or augment the biota already there. It is safe and effective, and it's also useful in the management of food allergies. So if you have food allergies, you should be on a good probiotic, and it'll make you more resilient and less apt to react to different foods and stuff. And so, really, that should be a treatment of choice. So if you've ever been told to eat yogurt and stuff, if you have a yeast infection, I would say yes, but avoid the really sugar-sweetened yogurt. So go with plain, and make sure it has live cultures, etc. in there, and that basically acts as probiotics. And you can also do fermented foods and stuff. Prebiotics, sometimes they're mixed in with the probiotics, are basically food for the probiotics or food for the good guys. So they're nutrients that will encourage the growth of your beneficial microbes, but that your harmful microbes don't like. So, for example, you have fructans. They can encourage the growth of bifidobacterium in the large intestine and discourage the growth of pathogens. So it's usually fiber of some sort, like psyllium husks and other things like that. They're soluble and insoluble fiber, and both have benefits that are various and stuff like that. So the difference between soluble and insoluble fiber is a soluble fiber, if you mix it in water, it will not thicken the liquid at all. It just will dissolve, and then you can drink it. And like acacia fiber is one of them. And then you just, it goes to your gut and it helps the good bacteria grow. The insoluble fiber, when you mix it in water, it gets thick. So some of the stuff with psyllium husk and all that is such. You always want to make sure you follow with lots of water. And if you are taking one that's an insoluble fiber. When you mix it with water, you want to drink it pretty quickly before it gets too thick. So our next case, the 30-year-old mission worker that came back with malaria, right, from working in Nigeria. So let's think about what challenges and stuff. So what would be one of the challenges in treating malaria and other protozoan infections? So what do you think there? So enter your thoughts there. And so your agents to treat protozoal infections, you have quinine. Yeah, so that's the principal treatment for malaria. It's been so for hundreds of years. And it comes from, I can't remember. Anyway, there's a natural component to it, right? It has been replaced by synthesized quinolones, chloroquine, and primaquine. And there are several species of Plasmodium, which causes malaria, obviously, and many stages in its life cycle means that no drug is universally effective. So you had the 4 species of Plasmodium, and some of these guys have developed some resistance to these treatments. So you have basically anti-protozoan, antimicrobial resistance. You have the problem of it has so many different life stages. So which one will be effective? Which life stage is it in? All of that kind of stuff, right? And the variety in the species and stuff. So those are all challenges. Some of our other cases, remember the GI symptoms case. The protozoans that can cause GI symptoms are your Cryptosporidium, Georgia lamblia, and Entamoeba histolytica. So again, this one's Cryptosporidium, Georgia lamblia, Entamoeba histolytica. And of those, you can take metronidazole. It's actually a good amoebicide, other than being an antifungal. It treats intestinal infections and hepatic disease that's caused by Entamoeba histolytica. And it can also be used to treat Georgia lamblia and Trichomonas vaginalis. And the other drugs with antiprotozoal activities are quinacrine, sulfonamides, and tetracycline. And then let's talk about agents to treat anti-helminthic infections. So we have, of course, challenges there because they're even more like us because even though they have unicellular stages, their eggs are just one cell, they are multicellular organisms also. So their physiology is rather similar to humans. So we're talking about treating flukes, tapeworms, roundworms, and larger parasites. If you block the reproduction, it does not affect the adult worm. So it might prevent them from making more babies, but it doesn't get rid of the adult worms, right? And so the most effective drugs do immobilize the worms or disintegrate or inhibit the metabolism of all the stages of the life cycle of the worm. So with immobilizing them, a lot of these worms actually hook into your intestines or hook whatever they're at. And if you basically paralyze them, they can't hook and hold on to, so they have to let go so they can be pooped out and you can get rid of them. So the next one, we're going to look at antivirals. So remember that was your 22-year-old college student that had fevers and all of that and ended up basically being a case of the flu. So again, put that in there. Which case was this college student? Did it have strep, RSV, flu, or Neisseria meningitidis? Did it have meningitis? And so it was virus. It was flu. And so some of the unique challenges of treating viruses. So for one, viruses rely on your cells, on your host cells for the mass majority of its metabolic functions. And so if you harm it where it's at, you're going to harm the host cells, so you're going to harm your own cells, right? So disrupting viral metabolism requires the disruption of cellular metabolism, so your cells. Measles, mumps, and hepatitis are all prevented through the use of vaccines, so teaching your immune system to attack and to control this. And AIDS, influenza, and a common cold, they all the tests for the need for more effective medications for the treatment of viral infections. We have not been able yet to develop a proper vaccine for all of those. And so the major modes of action of the antiviral agents is either they can bar penetration of the virus into the host cell, so they can mess around with the docking of the spikes on the receptor. They can block the transcription and translation of viral molecules. So transcription is the copying of the DNA or the RNA. Translation is making that into protein, right? And so if we block either of those processes, they cannot make more viruses, or we can prevent the maturation of the viral particles, preventing them from reassembling and leaving, etc. Draco is not as a theme from a character from Harry Potter, but Draco stands for double-stranded RNA activated caspase oligomerizer, and it is actually a breakthrough antiviral treatment that's currently being tested. So caspase are so many enzymes that can destroy different things, and it's being able to destroy double-stranded RNA. So that's pretty cool. And you can look into that as just a matter of interest, if you will. And so these are everything that we have against viruses. So the ones that inhibit virus entry are Fusion, that's used to block HIV infection. We have the Tamiflu and Relenza, which is amantadine and all its relatives. And so they block the entry of the influenza virus. And so again, they have to be taken early on in order to work. The ones that inhibit nucleic acid synthesis, you have your acyclovir and other cyclovirs, and those work against herpes viruses, right? And then you have your ribavirin. They work against RSV and hemorrhagic fever viruses. And then you have your AZT and all its friends here, DDT and DDC and all of that, and D4T. Those all work against HIV. They're target reverse transcriptors in HIV. And then basically everything else here is on HIV. So we have a few more here that rapine and stuff that are nucleotide analog reverse transcriptase inhibitors. And then you have some that inhibit viral assembly and release, and Indinavir and Sequinavir are those. And again, they just target HIV. So if you look about, the amount of drugs that we have, and you can see like basically 2/3 of them, pretty much half to 2/3 of them are targeted to HIV. Okay, and so this is the last thing. Enter your questions here, and I just realized I did not cover something specifically, so I'm going to go back. Y'all bear with me to this craziness of slides. Let's see where I'm at. Okay, I meant to cover this, and I did not, and I want to go back and talk about it for a second. Okay, this right here. So antibiotic drug resistance, what is conjugation, transformation, and transduction? So there's different ways that the plasmid gene can be transmitted from basically from buddy to buddy. Conjugation is what they call bacterial sex. Is the pili, there's a pili that's formed that's a bridge basically between the two cells. The plasmid is copied and then sent across. Okay. And then transformation and transduction, we have one is where having to do with the bacteriophage. So the bacteriophage virus that infects the bacteria, instead of picking up viral DNA in its reassembly, picks up the plasmid, basically the bacterial resistance gene, and when it leaves and it goes and infects another cell, instead of it has the plasmid instead of the viral RNA. So instead of giving it the viral RNA to the new bacterial cell, it gives it the plasmid, which is the resistance gene. And then the last way is when a bacterial cell is destroyed. So, through old age, or simply it's destroyed through the action of an antibiotic, all the plasmids and stuff are loose. and they're floating around, and these guys can actually be picked up by other bacteria from different species. So, random floating genetic material from a destroyed bacterial cell can be picked up by bacteria that are nearby. Okay, so sorry about not covering that right at that moment. You also have that part of an assignment. So hopefully you can just dig deeper into that. And so this is definitely long enough for a lecture. And so I hope that this was helpful to you. And if you have any questions, let me know.